Hello.
Morning.
Morning. Great. Great to see you.
Good to see you too.
Looks like Lalith and Paul are here, so and
Hello, Bradley.
hi, and Varshith. Hello. All right, so I guess we can start if Lalith wants to give an update. I know he was upset that he missed Friday's meeting, but
Yeah, so I actually had some visa processing things going on with me, so I wasn't able to join. So sorry about that. And yeah, I had a meeting with Dick where we discussed about the visualization parts of like how hypergraph can be done, and also he had shown us some work where how the cells have been exponentially growing in a log scale, where like cell types. So we had an interesting discussion, and I am and in the like the GSOC project, I'm actually working on the visualization tool where I can use the hyper.
looks like he dropped out. We'll have to wait. There he is.
Sorry, I got cut off for some reason. Yeah. So I was working on the visualization part of it. So I'm halfway done. So probably by this week, I'll probably able to show that, how you mean the way the neural networks learns, and the embeddings of the neural networks, the way it got learned, their hybrids, and we'll be able to visualize them.
Okay.
So I'll probably show that. So that's from my side.
All right.
And I'm actively looking into works where people have implemented C elegans and hypergraphs, and so that's the updates from my side.
All right. So you have the visualizations, the CL. So the people were doing hypergraphs and C elegans, or what was that last part?
Yeah. So I was thinking of reading the literature where people ever have tried about using hypergraphs on C elegans datasets, so that it could be a good benchmark. So I was looking into the research too. So probably if I can get to understand more in detail, like how the research has been working. If not, we can build the entire pipeline, and we can say that this is our standalone approach.
Yeah.
So yeah.
Yeah, I think I don't think anyone's really used hypergraphs. I mean, maybe, but
Yeah,
I know a lot of
it
people
was
have
tough
used.
to capture the literature.
Yeah, I know people have done things with like graphs in the connectome.
Mm.
So that's, I mean, and then the connectome, of course, isn't well characterized with regular graphs. Yeah, you might try looking in the literature for different, the way people have approached, the connectome, and look at how you can improve upon that. So, especially with respect to like function and how that could be part of the hypergraph.
Yeah, Bradley,
Yeah.
I consider this completing the C. elegans tree.
Okay. Yeah.
Okay.
Yeah.
It's kind of strange that nobody did it before.
Right.
Yeah. Sorry from the side.
They've had the data, but they haven't done it.
Right. Did you want to update us on the? What else is there to update? Maybe the centitial cells or the hypergraph? What? It sounds like Lalith and Dick had a meeting about the hypergraphs.
Yeah, I can give you something which I just discovered this morning.
Okay.
I've been concerned about that curve that I've got, which shows increasing intelligence over geological time.
Right.
Okay. And I read the book, the law. I forgot exactly the name. Laws of something. Book by Eigen. And I've gone through it, and he's concerned about population growth considerably. It's a 1975 book,
right?
And it's written in the how shall I say, in the modus vivendi of the Club of Rome people.
Oh, right, yeah.
Yeah. In other words, somehow we're going to solve this problem, and there isn't a word in the book about the pernicious sides of trying to cull people.
Right.
Okay, somehow we got to solve it. And they had 4 billion people on Earth at the time. Now we've got 8 billion, and people are panicking.
Yeah.
And they've forgotten the old UN projections that we would peak at 10 billion and then decline.
Right.
Okay, that's very inconvenient if you want to cull people.
Right.
Okay.
I'm not familiar with the Eigen book. What is that? Laws of.
Let me get it just a second.
Oh, okay.
Okay.
Eigen, Manfred Eigen and Winkler, Laws of the Game, the principles of nature govern chance. Yeah.
Yeah. The book was published in 1975 in German and later translated to English and Italian from an exponential before it blows up. Okay, they're very close if you try to fit the data. So I've got to go ahead and try to fit the data I've got, which is very limited, to a hyperbolic curve, and see if I can predict the singularity. Okay, the hyperbolic. In other words, the hyperbolic function has a singularity, and I don't know if it's the same as the Kurzweil singularity. Okay.
Yeah.
Okay, but they use the same word.
Right.
At any rate, I was trying to think this through, and I just came across something this morning, which is a group called nuclear genomics. And nuclear genomics, it claims that they can choose your embryo before it's born for intelligence. Okay. I don't know if that's correct or not.
Okay, so what is intelligence?
What is intelligence? I presume it's IQ. I don't know. I don't know how you test how you test an embryo for IQ.
Yeah, but someone could have an IQ and have abs and trip over their own feet all the time.
Oh, sure.
So what is intelligence?
Yeah, we have a name for that. That's oh shoot, what is it?
Absent-minded professor.
Well, yeah, that's one of them.
No offense to the professors on the call.
The other are there's a name for it. I think it's from the French. Somebody who is very intelligent in one small aspect of their life, and they're stupid otherwise.
Idiot savant.
Idiot savant. There we go.
Oh, okay, yes.
Yes. Okay. I mean, there are people like that. Okay. So I was figuring, okay, what if you did that for a number of generations? Would you make people that are smarter than the artificial intelligence?
Yeah.
Okay. So this is another way of getting to very high intelligence, at least a plausible path to it, other than making smarter and smarter artificial intelligence. Okay.
Yeah.
All you do is you keep doing this. Every generation you select for the most intelligent embryos. Okay.
Yeah.
I don't know if it'll work. I haven't tried. I can't say that the generation out of me is particularly intelligent, but okay. But then I didn't get to choose. So at any rate, it's an interesting way of, how should I say, manipulating genetics so that we get more intelligence. Now, whether more intelligence means more brain cells or more connections between them, etc., we don't know yet. Okay.
No, more Asperger's.
More Asperger's, right?
Yeah.
Right. Yes, I guess it's been proven if you make
neurodivergent.
If you make two Asperger's, you get more Asperger's.
Or two really intelligent people have a whole pile of Asperger's children. Yeah.
Anyway, of course, those people may be the very ones that are replaced by AI. So, I have a grandson who's interested in going into plumbing because AI will not replace plumbing.
Well, you never know.
At least until we get an artificial intelligence robot that can figure out plumbing. Okay. Whereas things like doctors, they're easy to replace. They're all being replaced by the current AI. So,
given that's my field, I actually think a lot of the capabilities of AI are very overhyped or overblown.
oh yeah, but that's because AI is in its infancy. It just made its breakthrough.
Well, the underlying architecture of LLMs, the transformer, means that AI will never have a true understanding of what it's doing. I mean, there's attempts to link it to like neurosymbolic methods. There's attempts with like retrieval augmented generation, but like even with RAG, it's still going to hallucinate references. So like, is something like that going to be reliable for making medical diagnoses? Unless we change the approach of AI ML, the answer is no.
Yeah,
I mean, essentially, transformer is a universal function approximator. So you're like, I think a lot of that's overhyped.
Yeah, I mean, I'm aware of the limitations of the current AI. For example, I was using Grok, and I did a search on Grok, and it came up with this flat statement that something wasn't true, and I pointed out that it was wrong. Okay, I quoted the source that proves it's wrong. So what it did is said it will modify its database based on what I told it. And that's extremely dangerous because I could have told it something false. Okay, and that's what I think that's one direction that AI has to be protected against, and that is people who are pernicious and put in false data.
Yeah.
Because AI will accept it. Okay, so that's an example of limitation of current AI also. It also sometimes makes up references you can't find on the internet. If you ask it for references, it just makes them up. And this is supposedly because the function of AI is not to inform you, it's to please you.
Oh no. I can think of a whole pile of ways that could go wrong.
Yes. At any rate, we're at the very beginning. I mean, I grew up with AI being a total flop.
Yeah.
There were a lot of people working on it, but the computers just weren't fast enough to do anything significant. So it was regarded as a flop for decades. And then all of a sudden, these deep learning things came out, and they're much better. I mean, I use it every day for reference finding, but with the caveat that every reference I'm given, I have to check. I think, given this nuclear genomics group and the idea that you could compound it over generations, at any rate, Varshith and Lalith and I are working on a paper that we're calling Brainy AI.
Okay.
And the idea of the paper is to try to figure out what it is about the human brain that's different from the construction of AI, and whether we can improve AI based on that. Okay, we don't have anything solid yet. Okay. However, we think we can do a comparison. I know none of us have supercomputers, but they're pretty fast, the ones we've got.
Right.
If we modify the AI, we ask the same question and just do better. Okay, so we think we have an objective way to test whether or not we're getting anywhere. Okay, so we kind of have to now collect properties of neurons, such as how many connections they have, whether they're electrical or chemical, how many cells there are, and we have to guess at how many different kinds of neurons there are, because one of the things is AI uses all neurons the same, as far as we can tell.
Yeah.
Are we right about that?
For what? All neurons are what?
All neurons that are simulated by AI or used by AI are fundamentally the same, except for the weights assigned to them.
Usually, yeah. I mean, you have things like spiking neural networks, but that's not usually used in a lot of the commercial AI. I mean, there are different ways you can do it. I mean, most of the ways are just kind of like they're units, and then the weights are the important thing. You take the weight matrix and
Yeah.
use that. Spiking neural networks generate like simulated spikes, and that's kind of what
Right.
in simulations of C. elegans as well. Although they
Okay.
don't produce true action potentials, but you can play with those, and you can actually get heterogeneous outputs. But that's it's kind of hard to work. They're harder to work with in terms of.
Okay. So
Yeah.
Varshith and Lalith, at least note that and see what literature you can find on it, or ask Bradley.
Yeah. Yeah.
Okay.
Also, I do want to mention I have a background in like a heavy background in AI/ML.
Yeah.
A few
Yeah.
peer-reviewed works in the field. So, to answer your question, I mean, activation functions can change, but the main way in which they're going to vary is the architecture of the layers. That's where most of the advent of neural nets, are.
Yeah, we just got a paper a few days ago on waves between neurons, so that whole groups of neurons can affect another group by a wave of some sort that goes through them, and that's a possibility. That book by Eigen says memory has got to be in the connections, whereas if there's ever a way of making memory to be in the RNA or the DNA, that might be an alternative. Okay.
Yeah.
So, there are all sorts of things to consider here while we're thrashing about trying to find what we call brainy AI.
No. Yeah. I don't know. We'll see.
Okay.
Yeah.
So it's fun, but this stuff this company's doing suggests a way of getting a smarter human brain, which just might exceed AI. But you got to breed for it, and you have to select for it. You have to do by deliberate selection.
Yeah.
And probably over many generations. I mean, the time scale on that graph I showed is the tick marks are at 100 million years.
Yeah.
And that's a lot of generations.
Yeah. It is.
Over. Okay, how do you keep the curve going? So we may be very dumb compared to 10 generations from now.
Yeah.
So I guess it's also a source of humility.
Yeah. Well. Okay.
Okay.
So yeah, Varshith is working with you on this. Could you introduce yourself?
In machine learning, and right now working on neural speech decoding from the neural activity. And I love speech decoding, and I'm interested in C. elegans too. So I got to know about this by Lalith. Yeah, Lalith is my friend here in Triple. And I'm really interested to learn. And recently I started working with Dick on Brainy AI.
Okay, it sounds good.
Yeah, let me show you that curve.
Yeah.
Because Jess, I don't think has seen it. Let's see, I've got to get back to here and start screen sharing. Screen there. Share. Okay, now in Word. Okay, can you see this?
Yeah.
Graph. Notice the scale. It's log 10 of the number of nerves, and these are for different organisms, and the age is given in hundreds of millions of years.
Yeah.
Wow.
And it's just coming up as an exponential. Look, this is an exponential fit to the data. Okay, now I made that exponential fit using Mathematica, so I don't know what the criteria are for the fit. You have to dig into Mathematica to find out. But this curve is going up, and it's not plateauing.
Well, so the log 10 is the number of neurons or nerves?
Nerves. It should be neurons.
Okay, yeah.
Yeah, I should reload that.
Yeah. So, I mean, it's like kind of like you have one example of like big brains or like these largely expanded brains, and that's mammals and probably most
Oh, yeah.
Yeah, like invertebrates, you have larger brains than say like in.
No, invertebrates are not on this. These are all regulating embryos.
Okay, they're all regulating embryos.
Well, yeah.
So
Yeah,
they're just basically.
If you make this curve for mosaic embryos, it's flat.
Oh yeah, I saw that one. Yeah.
Yeah.
So that was just the regulating.
Yeah, this is just regulating. So I figured the key to doing this, extrapolating the curve, requires understanding embryogenesis, but I thought that meant constructing an embryo, artificial embryo. So you see this reaction. There's two reactions to this. Wouldn't it be great to have a kid that's smarter than you?
Yeah.
Or no, that would make me nauseous.
Yeah.
Okay, so it's a curious thing, and they're actually claiming they're now selecting for intelligence.
Okay.
Okay, but they didn't think through if they have customers over many generations, then what? Because if it works, you see, by selecting for intelligence, we don't know if you're selecting for we don't know which property of the brain you're selecting for.
Right.
Okay. Whereas what we're trying to do is guess that for brainy AI. Okay. This because you're just selecting for intelligence, you don't have to know that.
Yeah.
Okay. And for what I can tell, for all the work on the brain, we really don't understand how the neurons work.
Well, how they put together intelligence, rather.
Yeah, how they put together things like intelligence, vision, etc. It's so we're hunting around for clues.
Yeah.
Okay.
Okay.
So it's kind of fun. It's now this is the opposite of what Elon Musk warns us all about, and that is that our artificial intelligence may replace all our jobs. Well, fine. Okay, let him see if he can get artificial intelligence to replace a sink tap, for example.
Yeah. So this is the story here.
I don't know. Well, you're in it. Yeah, there's also YouTube by this guy Khan Sadecki claiming that he's selecting for intelligence. Get that just a second. Let's see.
Paul has the story. Paul has the article up here. So yeah.
Okay, maybe you can. Here, let me get this first.
I just realized I was muted. I was trying to say I had the article pulled up. I can stop sharing if necessary.
Recently.
Well, you took that big banner out, so we'll use your version.
Yeah, that's.
I was asking if there's although I was muted, is there a certain part you wanted me to pull up?
Oh, no, the whole thing.
Okay.
If you can turn it into a PDF and send it to me, that would be great.
I can do that.
Okay, I'll put my email address in shortly. Just a second. Okay, okay, here it is. So, at any rate, it's kind of fun. I don't know if it'll make us smarter, AI, but we may find a path to it.
Yeah.
I think by starting with a dumb AI and seeing if we can make it smarter, one approach. I don't know. There's three of you there. Any of you know how to make a dumb AI that fits in a laptop?
Okay. Well, yeah. Thanks for that update. That was good. Look forward to seeing what you guys get do in that area. So
By the way, my answer is yes to that question, but it depends on what you're trying to do.
Oh yeah, play tic-tac-toe or something. I don't know.
I've literally trained LLMs on my laptop. So
Oh yeah, okay. Well, then with the code in the laptop, no internet
Yes.
connection.
yes, correct.
Okay, because we're worried about how much energy it takes AI to use up.
I mean, so I trained like a small parameter LLM. It took a hot minute. It took like 10 hours of training. So it isn't fast.
Okay, okay, slow learner.
Here's the thing. Even if you do like rent an A10 cloud instance, right? Generally these LLMs are so large that they literally train for days, if not weeks, even when they have access to hundreds of GPUs, quite literally.
Yeah. Yeah. So, at any rate, I'm not suggesting any of us buy a supercomputer, but
yeah,
at any rate, so that's what we're doing. We're trying to predict the future and ignore all the naysayers.
oh, okay. That sounds interesting, ambitious. Yeah. Morgan
Okay.
put a link in the chat. What is that, Morgan?
Well said.
This is CompuCell 3D's summer workshop is still going.
Okay.
Yeah. It's
Yeah.
I attended that last year. It's interesting.
Totally.
That type of modeling. It's not what I wanted to do, but it was interesting.
Right. What do they cover this year in the workshop? Is it like the same every year, or is it different?
Oh, it's the same every year. Yeah. He's got a very, it's a very comprehensive overview of the kind of different kinds of computational modeling that are possible.
Yeah.
Like certainly I caught some of the kind of traditional tellurium lib roadrunner kind of metabolic flux analysis that like you can find other courses that do that, right? But because he really tries to cover everything that is biologically relevant for somebody who cares about kind of virtual tissue modeling. He just covers the whole range.
Yeah, and they've been doing some really interesting things. Like in the last few years, I think after post-pandemic, they've been doing like a lot of interesting stuff with modeling different tissues and bringing out all the tools together.
Yeah. His Galim print is his immunological seminar series on Thursdays. I think it's Thursday.
Okay.
I don't know what Galim print is an acronym for, but the I is immunology.
Yeah, just those things. Yeah.
Okay, Bradley, let me make an analogy. We started DevoWorm because it was obvious that OpenWorm needed to understand embryology.
Right.
Okay? And I think the same thing is true about AI. We need to improve AI beyond its current primitive state. We've got to understand embryology.
Right.
Okay? And my reading of the literature so far is that in terms of embryology of the brain, that's extremely primitive.
Right. Yeah, there are a lot of people trying to incorporate elements of that, but it's kind of like it's kind of like they'll use a lot of analogies from developmental psychology, and they don't do it very well. But
yeah, I mean, if you look at AI and the simulated neurons that it uses, as far as I understand, they're very similar to the first model of a neuron, which is 75 years old.
Right.
Am I wrong about that?
Well, I mean, what do you mean by 75 years old? Like,
The first modeling of neurons was done 75 years ago.
okay, yeah. Oh, a 75-year-old model. Okay.
Yeah, yeah,
Yeah.
that's what I mean.
Yeah. Well, yeah. Well, it's
And that's what's used in AI.
right. Right. Well,
Okay,
it's an
I think
extremely
that's
abstracted version of a neuron. It would just be,
oh yes,
yeah.
yes, of course, the first model.
and language models are actually kind of using an updated form of that.
Right. Yeah. I mean, people, yeah, approach it in different ways. Yeah.
Well, just there are some differences in the transformer architecture.
Right. Right. But yeah, I mean, it's again like they're like, if you add detail to the model, it's like, what do you really need to add to make things maybe more efficient or more, maybe understand context. You have to really kind of define what you want in your model to have as an output to say, okay, this is what we need to add from the brain. I mean, it just doesn't automatically happen that if you mimic biology that you get. Yeah.
Yeah, I mean the brain it's got these waves. I've always wondered what on earth the waves doing. Maybe they're involved in intelligence. I don't know. There's all sorts of chemical things going on in the brain. I mean, your morning coffee is an example. Okay, so what does all this mean, and does it help in improving the intelligence of AI?
Right.
Maybe we should give AI caffeine. I don't know.
Well, you could do in neuroscience they use caffeine in some experiments as like they use different concentrations of the chemical. So it's like you could have like a model where the neurons are sensitive to some virtual chemical, and I mean they've done these kind of experiments in like agent modeling and artificial life where you'll have maybe like some sort of network where you have cells and then you give them like a concentration of a chemical and they behave a little bit differently. You could have like an activation function change or a difference in the way that the outputs operate or something like that. I mean, then you're getting into like applying a life to AI, and I know there are people working on that aspect of it, like doing things with evolutionary programming or different types of representations of one hot area, of course, is reservoir networks. Where, you have a bunch of neurons that are in this pool and they're connected randomly, and then you train the network and it produces like a patterned output because it's trained over it basically associates units get associated with one another and it outputs something. It's a very interesting and trendy area, but it's like it's very hard to understand like what's happening inside the black box. But my point is that you can use a lot of biological metaphors to sort of get to something better than what we have now. But it's hard to kind of know what that looks like, and people try these ideas and they get these ambiguous results, and
Okay, I got this triggered a question. You're familiar with computer simulations of murders of birds using Boids?
Oh yeah, Boids.
Yeah. Okay. Has anyone created such a simulation in which the Boids act like the birds that Alfred Hitchcock made a movie of?
No, I don't think so, but I mean.
And then we could have a real murder of birds.
Yeah. I know that. Well, I know Tom Porteges was working on some Boid simulations that had like where the birds could like see in front of them as well. Because the original Boid simulations were basically where you had these simple rules that every Boid would follow in parallel, and then it would produce these emergent behaviors. But what if you could like see things in front of you? Each Boyd had differential information, then what would that look like?
Okay.
You know, so I don't know. I mean, like, your answer to your question is probably no, but I mean, you know, there are ways you could probably implement something like that.
There we go. We should write a paper on the real murderous Boyds.
Right. It looks like Hussein's joined us. Well, Hussein, did you have any updates for us?
Hi. No, not right now. No.
Okay. Any other? Oh, go ahead, Susan. Do you have an update?
Yeah, I have an update. I just asked Dr. Sharif if I could get some optical coherence tomography images of salamander eggs. And he said, yes, as long as I got all the permissions. Like I have to make sure that I have insurance for his lab. I have to have the right permission for the salamander eggs, blah, blah, blah. I have to do all the paperwork. But he said that I could come into his lab and use his equipment if I could figure out how to make it work. So, well, new adventures.
This reminds me of the animal care committee that insisted that our axolotls had toys and that we keep them dry.
Yeah. Well, in my case, they're going to say, well, where did you get the salamanders? And I'm going to say from Kijiji.
That's pretty funny. I've seen people keep them as pets, both salamanders and the axolotls.
Yeah.
And they've shown me their toys. They show me what they play with. They're pretty fun creatures. So you would like.
Oh well.
Yeah, they do play with things, sort of. They're very slow moving.
maybe that's what we need. Zebrafish, a fast breeding, regulating embryo. We can try to select for intelligence and see if we can make a zebrafish that's smarter than the person.
Poor
Yeah, they're.
zebrafish.
I mean, the zebrafish's navigational capacity, based off of their proximity to objects and their ability to detect those things in a closed aquarium. That's one thing, you know, they often compare that to the human capacity for that too. So there's that, yeah.
I don't stick my head in an aquarium. Sorry.
At least when I studied zebrafish, when we looked, for example, for the autistic phenotype, how it's expressed in the zebrafish, we'd observe how they would prefer to remain stuck in one corner of the in one corner of the tank and observe their movements compared to that of the other one. So.
Okay.
Yeah. As for the embryo development of the zebrafish, it was interesting. I remember back in like 2017, back in 2018, that's when a lot of the labs at the NIH were able to produce very stunning images of how the embryo had been developing for the zebrafish too, especially getting the correct genes knocked down to light them up.
Yeah, well, as I said, you know, if we had an intelligence test on lower regulating animals, maybe we could test it without doing it on humans.
Yeah.
Also guppies. They reproduce fast.
That's true. Yeah.
Yeah. So I could give an update on the peptide project I've been working on with Dick and Savino's group over in Italy.
Okay. Yeah.
Remember we had shown images of these lattice structures, these hexagonal like lattice. Excuse me. Essentially, writing papers and answering questions on the formation of the lipid membrane, as well as the origin of life on and some very deep statistical and mechanical questions from, like, say, okay, excuse me, building on a lot of early ideas from Eigen and Winkler.
Okay.
And framing them yeah. Okay, so you guys are familiar. Eigen and Winkler had the book.
Yeah.
It's like laws of.
Yeah, we just talked about that book earlier.
Cool. Yeah, it should get in my mailbox pretty soon. Hopefully, laws of the game.
Yeah.
How the principles of nature govern, and I'm going to be sending it to Dick, and hopefully the Canada Post doesn't lose it. Yeah.
I found my daughter found a source where I could get the book online, but the online copy is defective. It has some pages duplicated, some pages missing.
Okay.
It was sloppily digitized, in other words.
That's unfortunate. Yeah.
Yeah. But I read what was there. So.
Excuse me. So anyways, Dick Savino and the colleagues over in Italy and I have been talking pretty thoroughly on the origin of life, stochastic versus deterministic origins, taking the ideas from the Eigen and Winkler book, Laws of the Game, and framing them in more so in terms of the modern stochastic physics. Whether or how homochirality and selection could arise from like the fluctuations, the breakdown of the breakdown of the law of large numbers. Okay, yeah. We talked about the fish ladder toy model with the Deemer paper,
right?
That's a more recent one. Yeah, and how we could model the small system world for a non-extensive high variance. Excuse me. So basically, when you're modeling membrane dynamics and the peptide clustering, you need to account for a lot of different factors, and one of those is what we would call the distributions of the fat-tailed fluctuation behavior, especially in the very, very early vesicle-like environments. Yep. You want to make sure you're modeling and simulating those correctly and accurately for the science itself. So we've been reading a lot of papers, some pretty damn recent, like the fish ladder model that Dick had published in 2023, and others dating back to like the 60s with like thermodynamics and similar ones. Yeah. The fish ladder model. Are you guys familiar with that? Did we ever talk about the fish ladder?
Ladder? Just give a brief review of that.
Excuse me, and I promise it's not like some climbing gate for fish to escape the aquarium. So the fish ladder is a toy model that you can use to Excuse me. Okay. Excuse me. So basically, there's this idea of this of a catchment area where the vesicles and other components sort of come together in a lake. Is that correct? Am I getting that right? It's okay.
Yes.
At thermodynamic equilibrium, when you have a large number of small systems, one of them would reach the threshold for life. Okay, and this is what you call a ladder, like climbing up to it. That's what it is, right? And we call it the fish toy. Yeah. Okay. The fish ladder toy model. So basically, what's the plausible null model that you can use to test the other hypotheses for the origin of life? It's Darwinian. It's Darwinian, yeah? I guess so.
No, it's not Darwinian until you have competition and selection.
Okay. Well, thanks.
Like that's the big step where everybody throws up their hands and say, "And there was life."
So yeah, fish ladder is like where the fish go upstream and they go up this structure where they just climb up into these different.
Yeah, it's based on a simple notion, and that is, a fish ladder is basically a set of small pools that are higher and higher as you go up whatever land is available to get to a source where a stream is coming.
Right.
Yeah.
Okay. And the idea is that at each point, there's a probability of the fish going up, and there's a probability that it ends up going down. So you can ask, given those probabilities, what's the probability of a fish ever getting up the whole ladder?
Yeah. It is funny because a fish ladder get upstream and get up dams. So that is not the fish ladder.
Yeah. See, if the fish gets up, then to succeed in getting all the way up are the ones that spawn.
Yeah. So each step is like an energy barrier when you go up.
Eventually, some of the fish will get up to the top of the stream.
Yeah.
Into the spawning area.
Yep. At least.
Given enough vesicles, at some point, one of those vesicles might reach the threshold for life. Yeah. Only a fluctuation.
So anyways, okay. So that would be the toy model part of the project. So we talked about the Eigen and Winkler, like the laws of the game, that introduced this this once and for all like principle. That was what it was, right? The once and for all principle from Eigen. It's called, right? The once and for all game. It's like this, like the birth and death process on the 8x8 board. We have a graph of statistical distributions of the pieces, and you see how they move. And you can see how when you start with like an equal number of like pieces for different players, the distributions would widen and become bimodal. So one player would win if they're competing over something. Yeah.
So do you have a visualization of that, or?
Yeah.
Yeah, to share your screen.
Absolutely, it's like I hate comparisons to chess. I think chess is just absolutely overrated. But like, it's like chess. Yeah.
Well, there are a lot of games that are like that.
Let me see if I can find it.
Yeah, this book is kind of weird. It's similar in a sense.
I don't have the book yet. That's the problem.
Oh,
yeah, I know.
Of it. Yep. I don't know. It should be in the mail sometime this week. Yeah.
All right.
I don't have the book yet.
So it's an 8x8 board or an 8x8 grid.
There's some analogy between their work and Wolfram's huge book on what was it? The new science.
Oh yeah, the cellular automata. Yeah.
Right. It's somewhat similar to that, except that this is stochastic, whereas Wolfram is deterministic.
Okay, I just googled this one. Yeah.
That's the game.
Oh, go ahead. Yeah. Sorry. It's not like NIM. It's not NIM. It's not NIM. But if you had a game you were playing in which excuse me, if you had a game in which two players controlled like different pieces or different sets of pieces, at one point there's this game. There's a version of this bead game called Once and for All. Excuse me. In which you need to get all of your opponent's values to your values by flipping them. Okay? At some point, I think it's like Othello. So it's like Othello, isn't it? Yeah.
Okay.
You guys know Othello.
Yeah.
That's the one where it's like, so you place pieces, and when you surround your opponent, you flip them all to your color. Okay.
Right.
Yeah. I think so. Laws of the game. It's called in. Excuse me. So the gist behind that, for what we are talking about here, is that oh, hold on, is this the book? Oh, it's only part of it. Yeah, I think the book was online. It's only part of it. Yeah. That's fine. How does a bimodal distribution emerge over time when you have some game that you can play by shifting your opponent's colors? Okay, like at some point, the pieces, most of them, some of them, all of them, would fluctuate back and forth between two colors or even other colors like red or blue or black or white or something like that. Yeah, and whatever bimodal distribution, you would observe selection dynamics in a game board like system. Okay, I think it's also interesting to see how the large scale birth and death processes would, this would basically align with the peptide raft simulations. Yeah. Okay. So fish ladder, like these once and for all game. I think it's called. Is that the name of the game? Once and for all. Is that it? Who was Winkler? I don't know Winkler.
I don't know either.
Ruthild? Okay, cool. Cool. Looks like it's Ruthel Winkler. Ruthel Winkler. Cool. Cool beans. Nice.
Right.
Oh, they're playing the game. They got it.
Yeah, looks like they're trying to. Well, the image you saw, it kind of looks like they're trying to model DNA or RNA evolution. Is that correct?
So it looks like Manfred Eigen und die Chemikerin, so that's chemist Ruthel Winkler. I don't know. This is Othello, isn't it? It looks like Othello. They got the black and whites and.
I can't see your.
Oh, you can't. I thought I was showing my screen.
You were, but not the image that you were talking about.
Okay.
I think you're on a different tab.
What's the difference between Othello and Go?
What is? Oh, well, those are two different games. Othello and Go are two different games. So what do you mean? What's the difference?
Oh, okay.
so Go is okay. So I mean, yeah, like the mechanics are different. Yeah.
Okay.
I think are they? Wait, no way. Go is way more complicated. Yeah.
Oh, okay.
Othello is like something I played in 4th grade when I was 10 years old.
Yeah.
And it's like, okay, hi guys, this has been fine. I need to get going to another meeting right now.
Okay. All right.
Yeah, exciting stuff. Like I said before, I'm really glad it's these things like these very simplistic games like Othello or perhaps Go, and I'm really glad it's not chess. I think chess is hella overrated as a model of game theory. It's not. Chess is stupid. Chess isn't about being smart. It's just about being good at chess. So
Okay.
That's my hot take. Everyone thinks strategy and game theory is all like chess. Nothing is like chess. Nothing in the real world is like chess. No.
Except a battlefield, because that's what it's modeled after.
You always see it in the movies and TV shows, like the smart characters. They'll be playing chess. I'm like, what are you doing? Why do you play chess? Chess has nothing to do with anything in life. Okay, that's my rant. But thanks so much.
Yeah. No problem, man. That's great.
Exciting stuff. I'm just reading papers and absorbing them into my bloodstream, to understand all the material and things. Dick, Savino, and I meet every other week. So we're meeting bi-weekly. Savino and his group, they have their obligations as well too. So we're just communicating and making sure we're all on the same page with things. Yep.
That's good. Yeah.
Thank you.
Well, thanks. Yeah. Thanks.
Thanks.
Yeah. So thanks for the thumbs up. And we had a couple of things in the chat here. Looks like Jess says I will be at the OGLAB summit at MIT later this year. OGLAB being different types. It's a lab that he's interested in there. I should have some means to share some related work from orthogonal DevoWorm. So there's a way to overlap or have something that would fit. Let me know. Yeah. So.
Yeah, I don't think I'll be working directly with Augmentation Lab, but they're sort of related to some other groups that I'm doing stuff with. Oh, you will be, Morgan. Okay. In person? Finally, a meeting in person. That's fantastic.
Yes, but you have to plug into those things on his head.
Yeah, right. I was already making kind of a Joe Pro social event around the Aug Lab, so that's really cool. I might have a few other folks. I mean, there's a bunch of stuff happening there. Yeah. So it's Augmentation Lab is a kind of a I don't know how to describe it. You can find out about them online, but I thought it was just going to be like directly, like let's physically, like kind of transhumanist, let's physically enhance our body in a direct sense. But it's actually much more broad than that. There's artistic stuff and some cognitive stuff and a lot of interesting things going on there. So I guess Morgan and I will be there, but I should have a means to present. Like I'm going to have like a few things to present. Like I got some new stuff and JoPro stuff, but also like if anybody here like kind of wants, I could do like a small like, hey, check out DevoWorm and this too. Like I don't know if it'd be like a lot of poster space or something, but just be able to promote a few things that we're doing here if anybody is looking for like an outlet in that kind of a space. But it should be exciting to do, and now will hopefully be a picture of Morgan and I that we can share here next time. So
yeah, the physical meetups. We haven't done that in a while, but
yeah.
you know, yeah, that's great. Thank you. Yeah, I don't know, Paul. Did you have anything you wanted to add, or?
I've got nothing at the moment. Quick question. So there's also Friday meetings?
Well, we have different meetings for different groups. So the Friday meeting is our open source meeting, and it's at a different link. And then we have our Saturday meetings for, actually, we do talk a lot about AI in that meeting, and that's also at a different link. If you're interested in those meetings, you can join. There's a different Slack to join. I can give you the link to that. And
yeah, that'd be great. I'm figuring out how I should contribute, and I've also got my own research ideas. So just trying to get myself oriented to the project.
Yeah, we might. Yeah, you actually, because you're interested in AI, you know, we might Saturday meetings might be really useful, or you could, you know, actually, I think develop some research there. So, well, I'll be in touch about that. Yeah.
All right. Thank you.
All right. Yeah.
This one's sort of like the bio-focused meeting.
Yeah, yeah.
Project-based.
Yeah. All right. Well, I think that's all for today. Thanks for attending, and see you next week. Good luck with everyone's different projects.
All right. Take care, everybody.
Take care. Bye.
Take care.
Bye. Okay, now I wanted to talk about something I hadn't gotten to in our regular meeting. And Dick asked a question last week about EvoDevo. And this is the evolution of development and the origins of EvoDevo. So I got some papers here that are very interesting in that regard. So from a theoretical perspective, EvoDevo is this approach that kind of came out of a number of findings that were happening in the late 70s and early 80s. So as people started to do molecular biology on developmental systems, people started to discover different experimental findings about the expression of different types of genes in the body plan, or what was known at the time as the body plan, and other types of findings regarding plasticity, and more importantly, that evolutionary relationship. So they were finding homologous genes and gene sequences that influence development. And by homologous, I mean in different taxa that are related by a common ancestor. So there was some similarity of function, similarity of structure. And then putting that all together, we get EvoDevo. Now, it's worth noting that there had always been a tension between sort of Darwinian evolution or theory of evolution and development. So notably, the German embryologists of the 19th century were very slow to take up a lot of the ideas of Charles Darwin. Some of that was because they were working in German and Charles Darwin was working in English. The other part of that was that conceptually it didn't really fit into what they were doing. So they were very slow to sort of accept evolution as being relevant to their work. And then you had other people in the history of in the 20th century who are like Waddington, who argued that there were missing pieces in natural selection and the theory of, especially when the neosynthesis was formed in the 40s and 50s. And so there was always this tension. And then, of course, in the 70s and 80s, we had these findings. And then it brings us to the sort of the theoretical synthesis that would result. And so this is EvoDevo. And of course, some people argue that EvoDevo isn't really a theory. It doesn't operate in the same way that, it's not necessarily as predictive as we'd like. It's largely a collection of findings, and then those findings have implications for evolution. But in terms of being fully integrated with, some of the ways in which we think about natural selection, namely that they're populations, and then we have population genetics to describe the population dynamics of different genes. That's not as prominent in EvoDevo. In any case, this is an article this is from Evolution, Education, and Outreach. This is by Brian K. Hall. This is on the past, present, and future of EvoDevo. So this was published in 2012, so it's a little bit dated, but it does give us a nice view of the history of EvoDevo, where it comes from. As I mentioned, this is something that is really kind of goes back to the 19th century, this tension between Darwin's ideas, Mendel's ideas, and the embryologists and what they were doing at the time. So he talks about the original paper here. So he talks about sort of the unique case of development. So the idea is that, we had sort of the process, we had the genes, and it was interesting that Charles Darwin actually argued for the importance of development in understanding evolution. But of course, the embryologists had their own sort of track of research and what they were doing. So it was a disconnect, even though there was this recognition that development was important. Interestingly, with Mendel's research and sort of his model of heredity in terms of genetics, people started to regard development as maybe not so important in understanding evolution, which was interesting because, for one thing, we knew about genes theoretically at that time, but we didn't really have good genetics techniques that we could actually look at the genes that were involved in development. So it was kind of a black box where we couldn't really get at the mechanisms for development, especially in terms of evolution. So it was kind of left on the shelf for quite a while. Now, in this paper, Paul says, research over the past 2 decades, this is basically the 90s and aughts when this paper was written, has opened that black box, revealing how studies in EvoDevo highlight the mechanisms that link genes, the genotype, the structures, the phenotype. This is vitally important because genes do not make structures. Developmental processes make structures using roadmaps provided by genes, but using many other signals as well. Physical forces such as mechanical stimulation, temperature, and the environment, in interaction with chemical products produced by other species. And, this is just kind of talking about all the things that are involved in developmental processes and how they come to bear on evolutionary change. Not only do genes not make structures or the phenotype, so, genes don't make phenotypes and morphological structures on their own. There are a lot of layers to this where genes are expressed. There are different actions of cells or different actions of the phenotype itself that go on to form these changes. But new properties and mechanisms emerge during embryonic development. Genes are regulated differently in different cells and places. Aggregations of similar cells provide the cellular resources or modules from which tissues and organs arise. Modules and populations of differently differentiated cells interact to set development along particular tracks. And organisms interact with their environment and create their niche in that environment. So a lot of these phenomena are couched under this term epigenetic. And of course, the term epigenetic is notoriously difficult because there's one set of meanings of epigenetic, meaning everything that is beyond the control of genes. So epigenetic is anything that maybe is involved indirectly in some process of expressing a gene or is the indirect product of expressing a gene. Epigenetic, however, also has another meaning, and it's very specific in genetics, which is that there are these epigenetic regulators or things like methylation tags or other types of epigenetic mechanisms that actually, we can look at them in much in the way that we look at population genetics. epigenetics. And so we can use both of these terms or both of these ways of viewing epigenetics and the effects of development to get a better handle on the evolution of development. So this paper kind of goes over a lot of this. So if you're interested sort of in the antecedents of Evo-Devo, there's this paper by McCain, 2009. She analyzed thousands of papers published between 1975 and 2004 and was interested in the impact of Conrad Waddington on Evo-Devo. And so Conrad Waddington, of course, was a developmental biologist who worked in about the mid-20th century. And he came up, of course, with epigenetic landscapes and some other insights. And so, but then, of course, he influenced a lot of the people later. And you have to remember, of course, again, that development was not something that we usually associated with evolution, at least not formally in the literature. So people were doing development, people were doing evolution. And then bringing those two things together was a challenge. Waddington, he made some conjectures about evolution, but not really working directly in that area. And then, of course, you had people who would use Waddington's work as inspiration for them thinking maybe more closely about evolution and, being able to bring those two things together. So there's really no founding figure of Evo-Devo. You could say maybe that Waddington was a founding figure, but Waddington never really coined the term, nor did Waddington ever really do a significant amount of work. It's synthetic work in bringing these two fields together. But the term evolutionary developmental biology appears to first have been used in print in 1983 by the zoologist and later environmentalist Peter Callow. So Peter Callow maybe is the founding father, so to speak, of the field, but this is not, this is, again, there are people working in the area. Maybe they used the term Evo-Devo and maybe they didn't, but I think there's a trend, though, you could say, from Waddington about bringing evolution and development more closely aligned. And so Callow was sort of a pioneer in this area, writing a number of books on sort of how do you incorporate things like life history or life cycles into different types of points of view. So he provided an influential treatment of life cycles from developmental, evolutionary, and physiological points of view. So that gave him sort of an in to thinking about development and evolution more closely, and then physiology being in a wide field that allows you to make a lot of these connections. Callow included a chapter on evolution and development, in itself unusual at the time, in his 1983 book, Evolutionary Principles, published by Blackie in their tertiary level biology series. Callow described the field of evolution and development as evolutionary developmental biology, noting that the area is a relatively new and complex one, so the reader should not expect to find fully comprehensive treatments in the literature. And so in this inaugural publication, Callow emphasized that natural selection could and did act at any stage in the life cycle of multicellular organisms, a conclusion reached by one of the founders of evolutionary embryology 109 years earlier. So there were precursor areas like evolutionary embryology, but again, this is kind of like the application of evolution to any field. It wasn't really that, it was kind of thinking about embryology first and then evolution kind of as a side. In his first paper on shark development, Francis Balfour argued for the role of selection in embryonic development as perhaps even more relevant for evolution than selection on the adult. So this was the work that was sort of the beginnings of evolutionary embryology. So this was a British person who worked on shark development or used this as sort of the model for what they were interested in. So this is from 1874. I see no reason for doubting that the embryo in the earliest periods of development is as subject to the laws of natural selection as is the animal at any other period. Indeed, there appear to me grounds for the thinking that it is more so. So an interesting kind of point here is that this is kind of mirroring the work of a lot of the comparative embryologists in the 19th century. So they would look at embryos of like chickens and humans, and they would notice similarities in those embryos. And of course, at the gross phenotypic level, you can see those similarities and you can say things about them. But at the genetic level, of course, they're very different. But as we found out, there are similar processes working across species that have a common ancestor that actually have this that provide, they're different, but they're also similar. So there are people like Stephen Jay Gould in 1977 who proposed these connections between development and evolution. You had Bonner in 1982, and you had other people who published things in the 80s through the teens, as this paper cites, as the roots of EvoDevo. And so, there were a number of different people who did seminal early work in this area. So Ernst Haeckel, of course, was one of these 19th century embryologists, and he had a theory that ontogeny recapitulates phylogeny. That is, as you go through the tree of life, you find that the developmental system is recapitulated. So when you can get evolutionary changes where you can get younger versions of other organisms that are related. So you can have evolution to juvenile traits across, over time. And so this is this theory was proven to be sort of disproven. And so, but the kind of idea persisted. But there's something about development that is relevant to phylogeny. You can see traces of the developmental process being moved around or sculpted. It's a common process that then is changed in different organisms. And that leads not only to changes in development, but changes in the adult phenotype in these different taxa that you see. So, you might have, say, for example, a developmental, as we would find out later, a developmental period that's expanded or a developmental period that's contracted. And that can lead to the changes in the growth of different traits. And so that's one evolutionary change we see. There are other changes related to ontogeny or development that involve changes in basically things like speciation or cladogenesis. And so, of course, all this was way beyond Haeckel. And, you'll hear often people say that Haeckel was proven wrong, which is true to an extent, but Haeckel's ideas persisted as these early observations. And so embryos provided a way to study evolution, but the theoretical framework was lagging by at least a century. So this is a good article if you want to learn more about some of these, the history of EvoDevo, not just the origins of the term, but like what people were doing in the 19th and 20th centuries to set up this theoretical advance. Okay, so that's the theoretical aspect. Now there are some seminal papers that kind of led up to this as well, because EvoDevo isn't just observing embryos and saying, well, yeah, they look very similar, so we can look at the phenotype and we can suss all the evolutionary details out. I mean, that would be nice if true, but we also have to have a more rigorous set of analytical mechanisms. And of course, we want to draw from genetics because, like as I said, you could observe commonalities in the phenotype, but the underlying genetics are very different. And so people started doing genetic studies in the 70s and 80s and finding these sort of principles for EvoDevo. So there are a number of bedrock cornerstone findings in EvoDevo. And a lot of times this forms a basis for the theory. So one of those bedrock ideas is that of Hough's segmentation and collinearity. So that is, you have this set of genes called Hough's genes, this family of genes that get expressed in sequence, and those set up different segments of the phenotype. So if you go to the fruit fly Drosophila, they have a number of body segments that are basically encoded by these conserved Hough's genes that are on the chromosome in order, and they're expressed in order, and they are conserved from evolutionary change, so they don't exhibit a lot of variation. In fact, it's essential that they be highly conserved in order to be functional. So this is the paper from 1978. This is a Nature paper. This is a gene complex controlling segmentation in Drosophila. This was E.B. Lewis. And this is just the original study where they discovered this gene complex and how it controls the segmentation in the phenotype. And of course, the segmentation happens in development. So you get the expression of these Hough's genes. It sort of sets up a coordinate system, more or less, for these different segments. And of course, if you've ever looked at an insect, you have these different body segments from anterior to posterior, and the order of these genes, and also, as they would later find, duplication of these genes can lead to changes in the phenotype, or sometimes preserving the same phenotype. So the abstract of this is that the bithorax gene complex in Drosophila, and bithorax means that it's two thoraxes, so bithorax. And so the thorax gets expressed twice, contains a minimum of 8 genes that seems to code for substances, controlling levels of thoracic and abdominal development. So this is where they have these segments in the thoracic and abdominal area of the insect. The state of repression is at least 4 of these genes is controlled by cis-regulatory elements and a separate locus called polycomb, which seems to code for a repressor of the complex. So you see, not only are they finding these genes, they're linking them to the phenotype, but they also have these regulatory elements and gene networks or gene regulatory networks that are involved. So a regulatory element, of course, is an upstream piece of DNA that basically determines whether or not some gene will be expressed. And then you get different transcription factors and signals from other genes in the genome. So there's a separate locus polycomb that is a repressor of the regulatory element and can repress the expression of the Hough's gene. The wild type and mutant segmentation patterns are consistent with an antero-posterior gradient. So this again is up and down the body with these different segments in the repressor concentration along the embryo, and a proximo-distal gradient along the chromosome and the affinities for repressor of each gene cis-regulatory element. So this is where we have this gradient in the phenotype, and this repressor concentration varies across the phenotype from front to back, and it determines how these genes are expressed. And then you have this gradient along the chromosome. So you have, again, collinearity, where these genes are lined up along the chromosome, and there's this effect of this repressor element that lessens as you go along the chromosome. So we're linking together genotype and phenotype. So all this is to say that there's this discovery that we have the segmentation in the phenotype, as you can see here. This is controlled by elements of the genotype. We can make the argument that, this is shared across taxa, across phylogeny, that it's conserved from a common ancestor. So, as we might expect, if we have major changes in this gene complex, in this group of genes, we can end up with serious defects in the phenotype. But we can also end up with these, what they call hopeful monsters, or large-scale changes in the phenotype. The likelihood, though, is that more often than not, this will lead to something deleterious. And so that's kind of the idea, that these Hough genes are a product of evolution, maybe conservation and evolution, and leads to something in the phenotype. You can tinker with the Hough genes a little bit and get different changes in the phenotype that represent changes across species. So that's the insight here. And then, of course, we have this. This is a figure here, figure 1. This just shows genetic control. So they worked out the circuit and how this works, and then how this works across development. So this is very nice work from 1978. And again, this was in Drosophila, but then, of course, they've worked on Hough genes in other taxa and in other parts of the tree of life as well. So another finding is that of conserved sequences. So like I said, conservation is a key concept in EvoDevo. You don't want to change genes too much that are involved in development, because if development is disrupted significantly, then the survival of the organism is disrupted significantly, affecting its fitness. So a lot of times you can have changes in development, and there are a lot of changes we're not going to get into, a lot of findings where you change the phenotype, like for example, if you change PAX6 expression in development, you can end up with a different number of eyes, and so that can affect the survival of maybe some organism, and it affects its ability to reproduce. So you don't necessarily see that phenotype propagated. You may also have developmentally lethal mutations. So sometimes, if these developmental genes get mutated, they can lead to non-viable phenotypes. So that organism can't even complete development. So this is a very important part of evolution because it speaks directly to reproductive fitness. This is a paper, this is a review article that leads to another original paper. This is a conserved sequence that sparked the field of EvoDevo. So the abstract of this paper, they talk about the homeotic genes in Drosophila, that these homeotic genes are conserved and utilized for embryonic development throughout the animal kingdom, so not just in insects or in Drosophila species, but also in humans and all throughout the animal kingdom. And it's worth saying that there's another analogy to that in plants called Madishetty genes. So Madishetty genes operate in a similar way to Hough genes in animals. But in any case, you see this sort of role of development. In a pair of back-to-back papers published in Cell, the journal Cell in 1984, researchers showed that the homeobox genes, previously identified as a sequence shared by homeotic genes in Drosophila, were also present in the genomes of diverse animals, so all across different taxa, across phylogeny. The first paper, which is McGinnis, 1984, and we'll see this in a minute, showed that genomes of both invertebrates and vertebrates, so invertebrates and vertebrates alike, contain sequences that cross-hybridized with Drosophila homeobox probes. What that means is that these homeobox genes that you see in Drosophila actually probably have their origins in the common ancestor of vertebrates and invertebrates. So this is something that's deeply conserved and has a deep phylogeny in animals. The second paper, Carrasco et al. 1984, identified a cross-hybridizing sequence in the model system Xenopus laevis. They then isolated the first vertebrate homeobox containing gene by cloning and sequencing the corresponding genomic regions. Xenopus laevis is a frog, so that's where we are in phylogeny. Finally, they showed that this gene, AC1, later named HOXC6, was expressed during embryonic development. The first evidence that developmentally expressed Drosophila genes could be used to isolate regulators of vertebrate embryonic development. So this is where we're looking in lizards and amphibians, and we're seeing this same type of arrangement, or this, homolog to the Hox genes we see in Drosophila. Now, it's interesting about these different studies is when people do these studies in a model organism, this or that, there are different nomenclatures for things. So a lot of times in genes, you have different nomenclatures. Sometimes they're homologs, sometimes they're paralogs, but they have to kind of figure out how these are related in terms of. Sometimes they'll use what they call like a homology analysis, where they see the similarity between genes in terms of sequence. Sometimes they'll look at how it's expressed and basically make the connection between two different nomenclature systems and how those genes have changed in development and evolution. So it's interesting how we work across these model organisms and have can make these findings. These findings led to a flurry of activity in the Evo-Devo field. So this was from 1984 forward, initially focusing on isolating Hox genes across diverse species, and then expanding to isolation in other gene families based on Drosophila orthologs. So orthologs are these genes that are homologous, and they retain similar functions across phylogeny, and they might have a little bit different sequence, but they do have this sort of common function. So we can actually use orthologs as well to look at sort of the common ancestry of these different Hox genes and things like that. This led to the notion of a conserved genetic toolkit for embryonic development, currently accepted, but unexpected at the time of its discovery. So this is a thing that really kind of is a key finding of EvoDevo, is this conserved genetic toolkit, meaning that it doesn't necessarily change a whole lot across phylogeny, and it provides us means to execute embryogenesis, execute a phenotype. That all being said, of course, the genome is not sort of the origins, or it's the ultimate origin of a phenotype, but it isn't the full contribution of the phenotype. And for that, you need to go to environmental effects and epigenetic effects, and that's another sort of theme in EvoDevo. We attempt to provide some context for the sea change in thinking that these discoveries brought about by referring to Jean Piaget's theories about the sequential action of scientific knowledge. So this sequential action and this idea of sequential acquisition and Piagetian theories of development is really an interesting parallel, because Piagetian theories of development are largely psychological, but you can see that sort of pattern of development or of acquisition in sequence. So you need to acquire things in a sequence in order to have them work properly. And of course, the sequence is enforced by the sequence and the genotype. But of course, if you disrupt that sequence, then oftentimes you end up with these developmentally lethal phenotypes. Okay, that brings us to our last paper. This is the McGinnis paper from 1984, or I should say McGinnis et al. And this is called a homologous protein coding sequence in Drosophila homeotic genes and its conservation in other Metazoans. So basically, it's these Hox genes that are homologous across species, protein coding because they produce proteins. And then this is in Drosophila homeotic genes, So it's a specific type of homeotic gene family. And then it's conserved in other metazoans or other animals. So what you see in Drosophila isn't necessarily exactly the same thing you see in frogs or in other animal species that have them, humans or what have you, but they're similar in terms of their sequence, and then also similar in terms of their function, but that's more general. They focus here on sequence, and that's kind of the point of the paper. So the summary reads, the homeotic genes, the Bithyrax complex, BX-C, and the Antennapedia complex, ANT-C, of Drosophila. So there is also the Bithyrax complex, of course, but there's also the Antennapedia complex, which deals with the antenna of Drosophila, and is a similar kind of system. Appear to specify the developmental fate of segments or parts of segments of the fly. So you have the bithyrax, or the thorax, the antenna area, and all of those operate by this homeotic gene. So the entire phenotype is organized in this way. We have previously reported weak DNA sequence homology between 3' portions of the Antennapedia and Fushi-Terazu genes of the ANT-C and the ultrabithyrax gene of the BXC. So Antennapedia and Fushi-Terazu are involved in the Antennapedia complex, and ultrabithyrax is involved in the bithyrax complex. Here we show that this DNA homology, or the homeobox, is due to a conserved protein coding sequence present in these 3 pattern formation genes. Thus, the functional homology between these developmental controlling genes is reflected in a structural homology in their gene products. So, in other words, you have this sequence similarity, you have a functional homology in terms of what it produces, and then this is reflected in the structural homology of the sequence of the gene products. So you have, I guess in this case, they're looking at similarities in the sequences of both the genes and the protein, and then that translates into the phenotypic similarity. The homeobox sequence is also present in a few copies in the genomes of some other invertebrates, and is even conserved in vertebrate genomes, including the human genome. Apparently, at least a part of these developmental switch genes from Drosophila are highly conserved during evolution and might perform an analogous function in many metazoans. So this is where we talk about the sort of way in which these genes are conserved, their sequences are conserved, the gene products are conserved, and then they play a role that's very much conserved in the anterior-posterior organization and other axial types of organization as well. And this just shows this in, this just shows how this is organized in Drosophila, showing some of these genes and where they're located in the genome. And what they mean by homeobox gene is that it's a box of genes in one location, and they're regulated through transcription factors, and they're all regulated kind of as a group, and as well as conserved as a group. A lot of times genes, if they're tightly located on the chromosome, they're conserved because they don't undergo, or they're much less likely to undergo any sort of recombination. And so this is a lot of molecular biology and sequence alignment. So I'm not going to get into that, but just to let you know that this is some of the ways in which they were doing this back decades ago, and they didn't really have the kind of tools we have now for this. So I'm not sure if this was. I mean, I'm really impressed with the kind. It's always impressive to go back to like the 80s and see how they were doing molecular biology, because it was not easy. Okay, so that's all I have to say about the origins of EvoDevo and some of the early findings of EvoDevo. All right, well, thanks for the original question, and I hope everyone learns something.